he was sitting in 1943 in his yard with his wife and his little daughter. And this was in Oxford during the Battle of Britain. And there was a bomber plane that crashed 200 yards from where he was sitting. And at this time, this young man had been, he was a tutor at Oxford. He was a very good teacher, and the students loved him. He was not a very successful scientist. He'd actually started to do his PhD, had submitted the work for his PhD. It was on cell culture and the growth of cells. And he thought that this work really didn't deserve a PhD, so he didn't go for the defense. So his thesis lies in the library at Oxford, which was never consummated. He never finished the PhD. And he was sitting there doing this tutoring during the week, and then this plane crashed. And then one of his close associates called him up on the phone and said, "Can you come to the hospital? One of our planes is down. The pilot has terrible burns. Maybe you can help us. You're the only person in Oxford who does any tissue culture. Maybe you can grow some cells from his skin, and this might help him." So the end result is that about 6 months later, this fighter pilot actually was discharged from hospital, but it wasn't because of what this young man, and this young man's name is Peter Medawar, it wasn't because he succeeded in his tissue culture methods or anything else, but this changed his life. He was here very involved in trying to solve this problem, getting this guy's skin grafts to work so that he would live. The patient did live, but he said, why is it that we don't understand how to transplant skin onto somebody who has this terrible third-degree burns? And he wrote a letter to the War Office in Britain during the war and said, somebody should study this problem of how can we transfer skin from one person to the other, especially when they have these terrible burns. So the War Office wrote back and said, maybe you can try to solve this problem out. Here's the surgeon who studies burns up in Glasgow. Just go and work with them and try to do something. So Medawar went and worked with the surgeon and studied one case of skin transplantation and came out with some theories of transplantation, realized the limitations of his studies on humans, and started to study transplantation in rabbits and then in mice. In the course of doing these studies, so now by now we've moved to the 1940s, he still doesn't have a PhD, he becomes a full professor, he studies transplantation, he describes the laws of transplantation, but nobody still knows about the cells that mediate transplant rejection. And then one of his students, Av Mitchison, shows that when you do this transplant, and they were transplanting tumors, they were transplanting skin, that the cells in the lymph nodes expand, and there are a lot of these cells in these lymph nodes that seem to collect. And he did the experiment, which then Medawar repeated. So Av Mitchison transferred these cells from the lymph node and showed that the cells from the lymph node transferred into another animal could cause a rejection of the tumor model he was studying. So even then, they didn't know they were looking at lymphocytes. They thought there were some cells in lymph nodes that caused rejection. So by then, and I don't have time to tell you the whole story, of course, Medawar had figured out the phenomenon of tolerance, and he went on in 1960 to share the Nobel Prize with Burnett for the studies on tolerance, after which he got some honorary PhDs. So he finally got a PhD. Okay? So this discovery of the role, and around the 1960s, Jacques Miller showed that the thymus was the source of the lymphocytes that caused this rejection phenomenon. And just before that, Jim Goins had shown that the cells in lymph nodes which recirculate in the body are these lymphocytes, and had shown with Medawar's help that these were the cells that mediated the functions of either making antibodies or causing things like tumor rejection. So we had then this picture emerging that everything wasn't mediated by antibodies, that there were these cells that could cause things like tumor rejection and do some other things like delayed type hypersensitivity and so on. So the big picture view, the first question I want to ask you is, what is the difference What is the fundamental difference in the biology of a T cell compared to that of a B cell? In terms of recognition, what is the fundamental difference? Which is better at recognizing things, and why? B cells are better at recognizing things. So then why are T cells any good? Big pardon? That's okay. Since I want to answer it myself at some point, right? So, but I want to see if you can think about it. Why did we evolve T cells? So again, you think of evolution, we evolved T cells and B cells 360 million years ago. That's when recombination started. But we did evolve an immune system 500 million years ago, which is a completely different immune system, which also has T cells and B cells. It's only found in lampreys and these jawless fish, basically. But in those animals also evolved T cells and B cells, and they had to do them together. So there's a need to have T cells. Although you can kill things in all different ways with antibodies, it was never enough. You needed T cells. Why did we need T cells? What's the difference? What's so special? No, I mean, that's too narrow a perspective for that. I mean, you need something bigger to tell me. Larger than life, like a bull. Yeah?
Because we were as carnivores or something like that?
Yeah, no. Nothing so, not about anything broader in terms of how we survive. Okay, so the first thing to remember is, as you pointed out, B cells see everything. And the strength of the T cell receptor is that it is virtually blind. It can almost see nothing. And that is a very powerful thing. To be unable to see things is actually a great virtue, because you learn, you get more efficient, you learn how to ignore most things. Okay? You don't bother with things. You don't have to be dealing with everything that's coming by. And that is the greatest virtue of the T cell, is that it has been taught as its basic recognition principle to ignore everything and to only focus on billboards that it can see, which are basically MHC molecules. Okay, so, and why this was so is you can think of a viral infection. So one thing that a T cell receptor can do is it can see inside a cell, which an antibody cannot do. Okay, because something from inside a cell can be presented on an MHC molecule, and so a T cell receptor can do it. But on top of that, even whether it was seeing inside the cell or not, you can imagine a situation where you have a virally infected cell spewing out humongous amounts of virus. Okay? An American term for it would be shitloads of virus. Okay? And you make antibodies, but the antibodies are distracted by the antigen because they can see it. So the antibodies should be coming here to bump this cell off, which is where we'll come to what an NK cell is supposed to do. Suppose you see the infected cell and bump it off before it infects all its neighbors. But the antibodies are so happy recognizing the stuff they can see that they don't reach the surface of the cell before it has infected 100 other cells. The T cell, on the other hand, cannot see a fragment of the virus, cannot see the whole virus, cannot see anything in the virus unless the virus has a fragment exhibited on its billboard, which is only going to be on a cell surface. So the T cell ignores everything going around and zooms in and focuses on the infected cell, and that's all it can do. And when it does that, it gets rid of this guy. So this is the big difference between T cells and B cells. B cells see everything, which is helpful in some circumstances, and T cells are blind to virtually everything. And this is why they're useful. And what the CD8 T cell does is basically to bump off its targets. So this is a CD8 T cell, this is some tumor cell, it's causing it to bleb off an apoptose by delivering the kiss of death. And it can do that so well because you're never going to see anything else outside the cell. Okay? So in this abbreviated lecture, we'll talk about CD8 cells in general. We'll talk about NK cells. And because the word NK seems so much like NKT, we'll talk about NKT cells, though they have nothing to do with NK cells. Okay? We'll just mention them, at least so you have a general idea of what they do. Okay? So you all know this. So CD8 T cells see class 1. So MHC class 1 is going to present peptides. What's the difference between class 1 and class 2? What kind of proteins, from where do proteins come for class 1? And from where do proteins come for class 2? So you know that CD4 is going to see class 2. Tell me the big difference, the defining difference again. Cytosol derived? Anybody else to take it? I'm not taking that one. Okay? Any other? You may be right, but I'm just asking you more specific answer than that. Say it again. You were all right, I'm sure. Proteasome. That's the right answer. Okay? It doesn't have to be from the cytosol. It could be from outside the cell. It could be from inside the cell. So all those rules are wrong. The correct rule is if it comes from a proteasome, it will go to class 1. If it comes from a lysosome, it will go to class 2. CD4 T cells will see antigens that came from lysosomes. CD8 T cells will see antigens that came from proteasomes. The proteasome might be seeing an antigen that was captured from outside the cell, brought to the cytosol, and then degraded in proteasome. Okay, so there are many different origins. The class 2 molecule might be seeing a cytosolic molecule that was captured by autophagy, got delivered to a lysosome, and then got delivered to class 2. So you can have proteins coming on different parts of the cell. In general, cytosolic proteins go to proteasomes. In general, membrane and extracellular proteins go to lysosomes, but it is far from absolute. Okay? So the basic principle is where did it come from? So CD8 T cells can not only kill, they can make a lot of interferon gamma, they can activate macrophages. They're very good at activating macrophages. They do a lot of things that Th1 cells do, and they do it rather well, and that's important. Okay, so main defense is against viral infections, but they can also protect you against bacteria and fungi. Immune surveillance of tumors, we all think about that today. They can be involved in autoimmune attack on self-tissues. It's not that you only get CD4 attack, you get CD8 attack as well. And clearly in rejection, both CD4 and CD8 are important, but CD8 is very important. Okay? So Abdullah has done this, so I don't have to spend time on it. He probably did the same slide. So basically, proteasome-derived peptides go through TAP and everything else, go to the cell surface. Okay, now cross-presentation, did Abdul cover that? No? Okay, so cross-presentation is when, let's say I have an infection in a brain cell, and my brain is virally infected. And somehow those brain cells are going to have to activate a T cell. But the brain cell doesn't have co-stimulatory molecules. It doesn't go to a lymph node. It's not going to be able to activate my T cells because it just doesn't have, and this antigen is only found in brain cells. But if that antigen from the brain cell could be picked up from outside the cell by a DC, ingested by the DC, so even if it came from outside the cell, there are DCs, and they are specialized conventional DCs, which have the property of taking proteins from the outside and then from within phagosomes or endosomes, transferring the protein into the cytosol. So the protein came from outside, transferring to the cytosol, allowed it to go to a proteasome, load up class 1, and activate a CD8 cell. And without cross-presentation, we wouldn't be dealing with viruses. For our CTLs, many of them against viruses are basically generated by cross-presentation. An apoptotic or dying cell is swallowed up by the DC. Proteins are chewed up. Some of them get thrown out into the cytosol, go into proteasomes, create peptides in the proteasomes, load up class 1, and they can activate CD8s. So even if I have an infection only affecting my liver cells, I can activate CD8 cells because my DCs can capture the antigen coming out of the liver cells and activate CD8 T cells. So even though it's not synthesized in a DC, it can activate a CD8 T cell. Okay? Stop me if you have a question about this. So cross-presentation is the key to getting antigens from outside a cell and presenting on class 1. CD8 T cells expand like crazy. You want a one-on-one interaction of a CD8 T cell with a target. So in an HIV patient, this has been demonstrated, that at the peak of infection, you can have 40% of all your T cells against a single clone. So using tetramers, you can go and look at specific clones, and you have these huge expansions. But after the expansion, you need to have a contraction. And so the T cells decline in numbers, and then you have memory cells that are left behind. Now, CD4 T cells help CD8 T cells in many ways. One of the ways they help is called licensing. Okay? And in licensing, what happens is that you may have a viral infection where there's not enough co-stimulation, but you want your DCs to activate your CD8 cells. So the viral infection might cause enough CD4 T cell activation. The activated CD4 T cells then tell DCs that, there's an infection going around, put on more B7 on your surface. So now the DCs have been licensed to become better DCs, even if they have triggered directly by TLR ligands and express B7 as a result of that. Helper T cells cause the DCs to be licensed. And then those licensed DCs can help a CD8 cell. So this is to tell you that CD4 cells can make cytokines that help CD8 activation, but CD4 cells can also license by inducing B7. This is the co-stimulator shown here on a DC. And so CD4 T cell causes the DC to have more B7 that allows better CD8 T cell activation and get T cell activation. So CD4 cells are drivers of CD8 activation often because of the licensing phenomenon. Now, the basic way in which CTLs kill is by making perforin and granzyme. So basically, you're going to have the T cell form a synapse with the target cell, deliver perforin and granzyme into the synapse. The perforin and granzyme are going to then enter the cell. Perforin is going to actually get into the cell, make pores and help get into the cell, make pores maybe within the cell as well. The granzyme is basically a protease that will activate caspases inside this cell and cause apoptosis. So you want the cell to wither away and die and go quietly into the night without inflammation. And the CD8 T cell does that, the CTL does that, by delivering perforin and granzyme into the cytosol. Okay? So you have this nice conjugate formation between the CTL cell and the target cell. You have this little cleft in between. Perforin and granzyme are delivered, and you get killing. Okay, so perforin and granzyme are the key killers here. The death is by apoptosis. That's all you want to remember from the broad means of killing. Now, a lot of pathology that we see in tissues. I've had hepatitis. I don't know if any of you have had hepatitis. I've had hepatitis and malaria, hepatitis A, of course. But when you have hepatitis, the reason for your bilirubin going up and you're having jaundice is that your liver cells are being killed in a good way. Your immune system is killing your infected liver cells. And the liver cells drop out because they're being killed, because they shouldn't be spreading infection. And as a result, you lose liver function. So when you see CTL-mediated injury, like in hepatitis, it's basically a good response trying to protect you, causing the liver to be damaged and causing you to have jaundice. Okay? So a lot of tissue injury that we see may be in response often to a virus, or when you mistake something else for being a virus, and then you attack your own cells, and the CD8 cells kill them. Okay? Now, Abul covered this, so I won't touch on it in any detail. So during a T cell infection, a CD8 T cell activation state, you do have often the phenomenon of exhaustion. And the reason you have exhaustion is that having a lot of T cell activation, a lot of CD8 T cell activation, as I just showed you in the previous slide, can cause damage. And you want to actually dampen that damage. And so a natural phenomenon that we've developed is that when we get a lot of CD8 T cell activation, we tell some of the T cells to quieten down and become exhausted T cells. Okay? So when you reverse exhaustion, like with PD-1 blockade, you can cause a chronic viral infection to go away. You can cause a tumor to melt, as has been very dramatically demonstrated in the last few years. Okay, so exhaustion is a state of when you have effector CD8 T cells, which are then told to quieten down and not be so damaging. Okay? And you can reverse exhaustion with PD-1 blockade, and you heard about this from a bull, so I'll go on. Okay, so I've covered CD8 cells. Is there anything else you want to know about CD8s? Okay, then I'll do an ode. Yes? Antigen must be expressed in MHC class 1 to activate CD8 T cells. Yeah, so in order to activate CD8 T cells, MHC class 1 has to present a peptide. The specific CD8 T cell sees the class 1 peptide combination with co-stimulation, gets expanded, and becomes a CTL clone. Okay? Any other questions?
You mentioned that CD8 T cells may also be effective autoimmunity, and I was wondering what an example of that would be.
So there is examples even in diabetes that you can have CD8 T cells going to the pancreatic lymph node, causing tissue damage. So there are a number of examples. There are cases of myocarditis where CD8 T cells are mediating damage. So you have to think about how did that antigen get presented? Was it by cross-presentation? Now, they're not the drivers. I would say that we still think of CD4s as the main drivers in those circumstances, but clearly there's evidence for CD8 T cells doing that too.
How do we know it's not a virus that they're going after?
Who knows what autoimmunity is? We don't know. I mean, they might have been going after a virus, but you can show, at least in some of these cases, you can show specificity for a self-peptide. So it may have been induced by a virus, may be cross-reactive against a virus, but I wouldn't think of it as the driving mechanism, but the phenomena exist. Yes?
Transplant rejection, how does the cross-presentation take place? Like, because the
Yes, in transplant rejection, you can cause activation by cross-presentation as well. But then, in transplant phenomenology, we call things direct activation, indirect activation, depending on whether it's MHC directly or not. But there's many phenomena. Yes?
current understanding of
Naive T cells exist. Okay, so the question is about the roles of naive versus memory CD8 cells. Naive T cells, naive CD8 cells, are like any T cells, exist to generate memory T cells, but most will die of unrequited love because they won't see antigen. Okay? When they become memory cells or effector cells, so the memory cells will be there to respond again, but it's the ones who do the damage are the effectors. So you make effectors, a small number of the effectors will become memory, most of the effectors will decline. There are memory cells that are created without any antigen. That's a different phenomenon. We haven't gone there yet. Okay? But yes. So a bull very emphatically told you that only protein antigens activate T cells. We are talking about CD4 and CD8 T cells. We're going to come to NKT at the end. Come back at 4, a bull. Okay? Yeah. Anyway, there's a break. Yeah. So in general, we only think of protein antigens with T cells. Now, there are some exceptions. I don't want to go into them. They are really rare exceptions. Which acts as an antigen? They do not act as antigens. Oligonucleotides that are being given are given as adjuvants to activate TLRs. They're not working as antigens. So SLE is not, in SLE, the nucleic acid is being seen, say it's in a chromatin complex. The nucleic acid may be recognized by a B cell. The protein in the chromatin complex may provide peptides for CD4 help. So the T cell is being activated by a protein antigen. Only when you have something that's a non-protein complexed with a protein are you going to get T cell help. Because MHC molecules present peptides. I mean, that's what you should take away. Any other questions? Yes.
Cross-presentation can be mediated by any APC or
So there are specialized CDCs which are very good at cross-presentation. They've been defined both in mouse and humans. They are the ones that do cross-presentation well. You can only demonstrate these things in vitro, right? So those are the ones that do them best. Okay? Now, there are reports that even PDCs can cross-present when they're activated and so on, but the best ones are still some CDCs. Okay, so I'm going to do a little ode for all the people who are not born and brought up in America. This will sound a little strange, but maybe you'll know this culture better than the Americans. Okay? It's called the Ode to Ben and Jen. My noble cousin expresses CD4. He is a veritable cytokine store. But the DCs come hither, and he's ready to go. His CD40 ligand, he just loves to show. My cousin's promiscuous hits on passers-by, kisses the wrong B cells, then tells them to die. The right BCR can give fast the slip, shuts off caspase 8 by turning on flip. I grew up a cell of the CD8 kind. I was naive and aimless, bored out of my mind. I wandered like a cloud from node to node. CCL21 determined my abode. Along came a virus, infected the liver. Hepatitis B can sure make you shiver. My DC friends each picked up a sliver, and in the lymph node, I started to quiver. Is this the feeling that Ben had for Jen? Or when Romeo saw Juliet again and again? Peptide in class 1, and then some B7. I leap in delight, I'm in co-stimulatory heaven. I'm no longer a boring CD8 cell. I'm a killer now, baby, a real CTL. No fleeting feelings like Jen had for Ben. Proliferate like crazy again and again. The liver beckons to me, and my brothers as well. We each form a synapse with an infected cell. With perforins and granzymes, the little clefts we fill. Oh, what passion to lock lips but kill. Passion we have now, but once we did grieve. For siblings lost, forced to take their leave. They perished at the double positive stage. Self-structures they did strongly engage. They made some BIM and then felt weak. Their mitochondria started to leak. Cytochrome C bound to APAF1. Caspase 9 began to hum. We wept, but they chose not to fight. They vanished quietly into the night. That's all I know about CD8 cells. Okay? Thank you. So I'm going to briefly touch on NK cells and NKT cells for a second or two. So NK cells you should think of as the innate version of CD8 T cells. They exist largely to deal with damaged cells and virally infected cells. They can see a lot of other features of damage. One of the ways that they are going to deal with virally infected cells is to see antibody-coated virally infected cells and then bump them off with perforin and granzymes. They're going to form synapses to see the infected cell, deliver perforin and granzyme, and kill them. So they're often called this third lineage of lymphocytes, important against viruses, bacteria, and tumors. But remember, they're innate, they don't have that much diversity, and they're sometimes not effective. So they can also make cytokines, they can make a lot of interferon gamma, and they can also kill abnormal cells. So NK cells can sense DNA damage, they can see transformed tumors, they can see some cytoplasmic microbial infections, they can see other things, okay, the viral infected cells and so on. So if you have a stressed cell, for instance, or an antibody-coated infected cell, the healthy cell became these cells. NK cell can recognize these, discriminate it from a healthy cell, and then bump these guys off. Okay, so that's the job of the NK cell, is to look for infected and damaged cells and bump them off. So NK cells develop from a combined progenitor of both NK cells and T cells in the bone marrow. The thymus is not required for the development. They don't rearrange any genes, so they're not like T cells and B cells in that sense. We still call them lymphocytes. If you look in the peripheral blood, they look like lymphocytes, and there can be quite a few in the peripheral blood. And they're found in the spleen, in the liver, and even in the placenta. Very important, the placenta. So they can do a number of things. They can kill naturally. So they see something that shouldn't be there, they can bump it off. So they have receptors to see stuff that is stress related. They have receptors. They have an FC receptor, a specialized FC receptor, that can recognize an infected cell coated with antibody. We've called this ADCC before. They also kill just like a CD8 T cell does using perforin and granzyme. They also can make gamma interferon early, and they can make a bunch of other cytokines. Okay? So the function, now if you think about it, a viral infection is going to cause PDCs and other cells to make type 1 interferons. And those type 1 interferons are going to very nicely activate NK cells. Okay, so you make type 1 interferons, they can cause the death of infected cells, but they can also induce activity of your NK cells. IL-15 is a cytokine that is used by CTLs as well, which is also useful for NK cells. Some of these guys that activate Th1 cells can activate NK cells to make interferon gamma. So all of these, the similar functions are being broadly mediated by Th1s, CTLs, and NK cells at different stages of an infection. And IL-2 can induce proliferation and so on. Yeah? So the big picture, we looked at this before. You have a cell with surface antigen, antibody coats it. The antibody-coated cell, which is an infected cell, is seen by the Fc gamma R3, the receptor on NK cells. This receptor causes the activation of the NK cell. It releases perforin and granzymes and causes apoptosis of the infected cell. That is one of the major functions of NK cells. Now, one of the most interesting things about NK cells that was discovered was that they like to kill cells that lack class 1. Viruses like to get into our cells and shut down class 1 to prevent CTLs from killing them. And NK cells like to see cells which lack class 1. Okay, so I've gone ahead a little too fast maybe. Okay, so the way it works is that if on an NK cell you have a lot of MHC class 1, okay, even though you have an activating ligand, let's say this is a stress-related ligand, the NK cell will be inhibited by the MHC class 1 because it has an inhibitory receptor, or many inhibitory receptors, that the MHC class 1 will cause this NK cell to become ineffective. Okay, so if there's MHC class 1 on a target cell, it will inactivate the NK cell and won't do its thing. So if you have an NK cell which is seeing a lot of this activating ligand with very little of class 1, it will get less inhibitory signals, more activating signals, and now the NK cell will get activated. If you have only class 1 and if you have very little activating signal over here in green, again, you can get activation because this cell has no class 1. So it's the balance of the activation signal, which is good for NK cell activation, to the class 1 molecules, which are bad for NK cell activation. You have too much class 1, it's not going to work. It will be described in 3 slides, which will start from the next slide. Okay, I'll get to it. Basically, an inhibitory receptor. So, an NK cell has activating receptors and inhibitory receptors. So, in humans, we use KERs, some of our inhibitory receptors. They are killer like Ig domain containing receptors. So, you have inhibitory receptors and activating receptors. So, let's say this is the ligand for the NK cell over here. It's going to activate the NK cell. But at the same time, if there's plenty of class 1 on the target cell, the inhibitory receptor will be activated, or inhibitory receptor will signal. The inhibitory receptor brings phosphate. It has item motifs. If you go back to items and items, so the activating receptor is trying to activate kinases and activate the cell. The inhibitory receptor is recruiting a phosphatase. And the phosphatase is preventing the kinase from putting the phosphate on and causing inactivation of the activating signaling. Okay, so the basic way in which this is working is that there's enough class 1 on the target cell, the inhibitory receptor kicks in and dampens the activating signals. The NK cell doesn't get activated. It becomes an innocuous cell and doesn't kill the target. Okay? There are inhibitory receptors. There are many different inhibitory receptors and activating receptors. This is just some examples of the inhibitory receptors. I'm not going into all the details, but HLA-E is a class 1-like molecule whose main function it is to induce inhibitory signaling by NK cells. Okay? And HLA-E and HLA-G are used in the placenta to prevent the mother from destroying the fetus, basically by inhibiting NK cells in the decidua. Okay, so NK cells must be dampened, and that's how you prevent fetal rejection, in part. And there are a lot of activating NK receptors, and the one that we talked about before was Fc gamma R3, which I'm not sure if I have on the slide. Okay, so that's one of the important receptors. But this is it. So this is CD16 is Fc gamma R3. So this is the one that will recognize an IgG-coated cell, and some of the other receptors can recognize different things, like this NKG2D homodimer can see proteins that are expressed on the surface of a stressed cell. So you have a bunch of activating receptors. They basically see stressed cells or virally infected cells and allow them to be disposed of. So again, this is telling you a little more about the same thing that I've mentioned before, that the activating signals allow kinase activation, and the inhibitory signals. So if you have a lot of kinase activation and very little inhibitory signaling, you will cause NK cell activation. But if you get, and how that is going to work is you're basically going to cause the injured cell to be apoptosed. So injured cells and infected cells are going to be disposed of by apoptosis by perforin and granzyme mediated mechanisms. Now, although NK cells kill infected cells and damaged cells and stressed cells, that's one of the main functions, they can also cause macrophage activation. So think of them like Th1 cells, again, good producers of macrophage activation events. So you can have a macrophage which makes some IL-12 because it's infected by an intracellular pathogen. The macrophage can activate Th1 cells, CD8 T cells, and also NK cells. And the NK cells are like an innate cell that can make a lot of interferon gamma, reactivate the macrophage can now chew up the phagocytosed microbe. So it's important to realize that it does a little more than just killing, in spite of its name. So there are humans. So in mice, of course, we know NK cells are very important. You can do knockouts. There are humans with some very rare mutations in genes that either cause the absence of NK cells or the failure to have active NK cells. And those mutations have also taught us that these cells are extremely important for dealing with viral infections. So you have severe Epstein-Barr virus infections in people who don't have functional NK cells, also severe herpes virus infections in these guys. And if you deplete NK cells, this is of course in animals, you can enhance viral synthesis, enhance hepatitis, and so on in vivo. So in mice, you can show that taking away NK cells causes more viral infection. So there's a very important role of NK cells in viral infections. So NK cells are very important if you do bone marrow transplantation or basically hematopoietic stem cell transplantation. We all know that you have important effects of host NK cell mediated rejection of stem cells. In leukemia patients, so when normally you're worried about graft versus host disease when you do a bone marrow transplant, but in the case of a leukemia subject, you want to have a little bit of a graft versus leukemia effect, and there the graft versus leukemia activity can come in part from donor NK cells. Okay? So there's an importance clinically. Now, that's all I'm going to tell you about NK cells. And in the last few minutes, I will tell you about NK T cells. And what you want to remember is the names sound similar, but these are very different cells. And NK T cells, since many of you have brought up this question about lipids and non-protein antigens, NK T cells are a variety of CD4 T cell that is not a classical CD4 T cell, but recognizes lipid antigens. And it recognizes lipid antigens, which can be from microbes, can even be self-antigens, presented on a unique type of MHC molecule called, it's a class 1-like molecule called CD1. Okay? So NK T cells are a subset of CD1 restricted T cells. Okay? So these are T cells that are not seeing regular class 1, those are CD8 cells. They're not seeing regular class 2, those are CD4 T cells. These T cells, small in number, are actually seeing CD1 presenting lipids. CD1 molecules they look like MHC class 1. They have beta 2 microglobulin, but they have a very greasy groove, and they can bind lipids, and they can present lipids in the groove. And in humans, we have a bunch of CD1 molecules, and the cells that we think are really called NKTs are the ones that see CD1D presenting lipids. Okay? So NKT cells, and I'm using this now to imply what some people always call invariant NKT cells. Okay? So they express an invariant T cell receptor alpha chain. That means every NKT cell has the same alpha chain, same rearranged alpha chain. They are all going to recognize lipid antigens presented by CD1D. So in the thymus, when a cell is becoming an NKT cell, it is recognizing a lipid antigen as a double positive T cell and then becoming an NKT cell because it got selected not by class 1 or class 2, but by CD1 presenting a lipid. And that converted a small subset of cells into an NKT cell. In humans, this is always a V alpha 24 T cell receptor. So the T cell receptor is always of this kind in the invariant NKTs. In mice, it's different. Okay? And in humans, we call NKT cells as a subset of CD1 restricted T cells because there are T cells that can see CD1, B, C, and D. Yeah? In humans, there are many non-NKT CD4 T cells that can see CD1, A, B, and C. And these can respond, some of these can respond to lipid antigens from things like mycobacteria. Okay, so these are also seeing CD1s, but we don't call them NKT typically because they're not with that invariant T cell receptor. So that might be an arbitrary definition. So there's a small number of NKT cells in blood and lymph nodes, small number in the thymus, the spleen, and so on. A huge number of them in the liver for some reason. And they can be CD4 positive, they can be CD4 negative, they can be CD8 negative. So they're usually either CD4 positive or double negative. They have neither CD4 nor CD8, or they have CD4. Okay, that's how they are. They generally evolved from CD4 T cells, but we think that because they have the double positivity, they're both CD4 and CD8, which saw a lipid antigen and then went into this direction. In humans, they may not be as important as in mice. So there's a huge literature on NKTs in mice where a lot of important functions have been described for NKT cells. I think we need to be a little cautious. They're probably important in humans, but you don't have enough data to say that. Okay, so they recognize lipids. One of these lipids that people have studied a lot is alpha-gal-Ser. It's a ceramide, which can bind to CD1. And we're just dividing our NKT cells and non-NKT cells. The ones that see CD1, A, B, and C, we're calling non-NKTs. The ones that see CD1D and lipids, we're calling NKT. And this may be guys that see microbial antigen, or they might see self-lipids. Okay, so there's a thinking that sometimes you activate an NKT cell seeing a self-lipid because that NKT cell gets activated by a DC that turns on the NKT cell, but sometimes the NKT cell is actually seeing a microbial lipid which is being presented to it, and there's a whole pathway by which presentation is going to occur. Okay, and my last slide is, so NKT cells are thought. So we put NK cells as innate. We put CD8 plus T cells and CD4 plus T cells as being adaptive. And NKT cells are among a group of a small number of cell types that we think of being somewhere in the bridge between innate and adaptive immunity, which can serve amplification functions and can make lots of cytokines. Okay? Now you can ask me questions on anything at this point, because I've brought it back to being on time. And if something wasn't clear, let's go back to it. Okay? It won't be the order, I'll do a wrap. Questions? Yes. Yeah, so the question was about if you reduce MHC class 1 expressions, will NK cells come into play? Generally, they will try to come into play in those circumstances, but some viruses have been clever enough to make decoy class 1 molecules and shut the NK cell off. So, viruses are smart. So there's many mechanisms of which the NK cell is being overcome. And so CD8 T cells they're trying to overcome, but we try to get around it. We make more interferon gamma, we activate a little bit more of the class 1 molecule, get a little bit in with some peptide, that might be enough. But NK cells can often be inhibited too. But you're right. In general, if you downregulate class 1, NK cells are more important then. Yeah? Any other questions on the vast world of B cells, NK cells, NKT cells, CD8 T cells, or anything else? Or different types of DCs? Okay, so I'll summarize everything, and this is the only way I think you can remember immunology, if you want to keep it in your mind forever. I actually remember some things only from some other processes. Okay? So this is the lymphocyte wrap. Pathogens lurking everywhere. Go get them first. They never fight fair. Give me a D, give me a J, put a V in frame, it'll be A-OK. Surrogate light chains, pre-TCR, kappa or alpha can't be too far. Thread that peptide into TAP. Everybody do the lymphocyte wrap. Naive cells dance from hall to hall, pining and hoping to get that call, looking for antigen below and above. Many will die of unrequited love. Thread that peptide into TAP. Everybody do the lymphocyte wrap. DCs lurking everywhere. They'll get your antigen, don't despair. CCL21 to CCR7. Come in, DC baby, make this lymphocyte heaven. Thread that peptide into TAP. Everybody do the lymphocyte wrap. Wake up T cell, activate ZAP. T cell says, takes 2 hands to clap. Give me signal 1, give me signal 2, and I'll make you a cytokine brew. Thread that peptide into TAP. Everybody do the lymphocyte wrap. Collectins, complement, macrophages. That's what you get in the early stages. Then antibodies, cytokines, CTLs too. Hey, pathogen, that's the end of you. Thread that peptide into TAP. Everybody do the lymphocyte wrap. We're pretty much on time.